In this module, we will talk about the diseases that affect the sugarcane. The first disease that we talk about is called red rot or lal kuch. The casual organism name is Collatotrichum falcatum. The nature of damage that it causes is it causes the sugarcane to split. It is the most common disease of sugarcane and causes severe loss of sugarcane yield and quality. It affects stalks of sugarcane, leaf midribs, and planting material. The perpetration is primarily through infected sets or canes and also through diseased stubble or debris, and also to some extent by resting propagules in the soil, as it does not survive in soil for more than 5 to 6 months. With the advent of the pre-monsoon showers, symptoms of the diseases start appearing. That is, drying up of the third or fourth leaf of the crown at margins. Later, the entire crown dries up and drops down. Brown or reddish-brown stripes appear externally at nodal region. On splitting, the internal tissue becomes red with white transverse bands. With onset of monsoon, whether the weather is most suitable, full manifestation of the disease takes place. Favorable conditions are a mean temperature of 29 to 31 degrees Celsius, drought conditions during the initial growth phase, high atmospheric humidity more than 90%, waterlogged conditions, continuous cultivation of the same variety in the same field. Just to summarize, red rot is noticed by the drying up of the leaf, crown at margins, followed up by drying up of crown and falling off. Appearance of red and brown stripes at the nodal region and internal tissues becoming red with transverse bands. To manage red rot, we need to use red rot free sets or planting material. Then we need to treat those sets by putting them in a water solution of water with trophy at the rate of 3 gram per liter. After 1 month of planting, we again spray the crops with trophy at the rate of 3 gram per liter. The name of the second disease is smut or Chabukkani. The casual name is Ustilago scitamine. The nature of damage that it causes is it causes the growth of sugarcane to stop, and it also causes the affected cane to die. It is a major disease in sugarcane and is observed on almost all varieties of sugarcane. The disease can be identified by the production of a flagelliform appendage, a black whip-like structure from the central core of the meristematic tissue. It affects stalks of sugarcane, leaf midribs, and planting material. When the lateral buds begin to sprout, they may produce whip-smutted side shoots. The perpetration is primarily through infected sets or canes, diseased stubble or debris, wind-blown spores, and to some extent by resting propagules in the soil carried by the irrigation water. Favorable conditions are high temperature in April to May, dry weather, and acute shortage of water leading to high incidence of the disease. Ratoon crops are more damaged than the planted crops. To manage smut, we need to use smut-free planting material. Then we need to treat those sets by putting them in the water solution of water with trophy at the rate of 3 grams per liter. After 1 month of planting, we again spray the crops with the trophy at the rate of 3 grams per liter. The next disease is called wilt or marrog. The casual organism is called Fusarium moniliforme. The nature of damage that disease causes is it affects the cane to wither and then subsequently die. It is an important disease affecting sugarcane. It is observed to be endemic in most of the sugarcane growing areas. The disease symptoms first appear during the monsoon and post-monsoon periods. Affected plants display stunting and an unhealthy appearance, followed by yellowing and withering of the crown leaves. On splitting the canes at early stage of infection, diffuse purple or muddy red color in conical patches can be seen at each node just above the ground ring that pass through the one internode from the other. At later stage, spindle-shaped cavities tapering towards the nodes develop in each internode. Sometimes cavities also develop in the internodes. The perpetration is primarily through infected sets or canes, diseased stubble or debris, wind, rain, and irrigation water. Favorable conditions are overnight temperature above the seasonal averages, cloudy weather, misty weather conditions, and lack of air movement. To manage wilt, we need to use wilt-free planting material. Then we need to treat the sets by putting them in a water solution of water with Mangala COC at the rate of 2.5 gram per liter. And after 1 month of planting, we again need to spray the crops with Mangala COC at the rate of 2.5 grams per liter. The next disease that we talk about is called a grassy shoot disease or Gaftalwad. The casual organism for which is called phytoplasma. The nature of damage that it causes is, it causes the plant growth to become stunted. It is an important disease affecting sugarcane throughout its growth phases, and it is observed to be endemic in most of the sugarcane growing areas. Ratoon crops are more adversely affected by it. In this, the plant growth is stunted and does not develop into a whole plant. This disease causes production of numerous small and thin tillers having narrow leaves with or without albinism. Varying degrees of loss of chlorophyll, ranging from total green to white, premature and excessive tillering gives a crowded appearance like grass to the clump. The disease infestation can be managed by the use of disease-free planting material. That is, you choose the sets which are free from grassy shoot disease, and roguing of the affected plants. The next disease is called Polka Boink. The casual organism for which is Fusarium moniliforme. The nature of damage that it causes is it stunts the growth for sugarcane, and it also creates distortions in the growth pattern. This is a rapidly emerging as a major disease affecting sugarcane and is observed in most of the sugarcane growing areas. The plants become stunted and there is distortion of the plants. The base of affected leaves is often narrower than of the normal leaves. Chlorotic areas at the base of young leaves followed by distortion, which is wrinkling and twisting and shortening of affected leaves. The perpetration is primarily through air, and secondary infections are through the infected planting material, splashed rain, irrigation water and soil. Favorable conditions are drizzling rainy days and a temperature of 20 to 30 degrees Celsius. The way to manage the disease is, we need to buy the disease-free planting material, and then we need to treat these sets by putting them in a water solution of trophy with water at the rate of 3 gram per liter, and after 1 month of planting, we again spray the crops with trophy at the rate of 3 gram per liter. Thank you.